So we are now talking about the lactose operon. So lacoperon or lactose operon consists of a regulatory gene which has been given in the figure as I for inhibition. It also consists operator gene denoted by the symbol O, letter O, and three structural genes Z, Y, and A. Beside these three genes, there is a promoter site denoted by P next to the operator gene where the enzymes RNA polymerase would bind. The structural gene Z, Y, and A respectively code for the enzyme beta galactoside, galactoside permease, and galactoside acetylase. Normally, beta galactoside hydrolyzes the lactose if present in the medium and break down this lactose to form galactose and glucose, while galactoside permease responsible for transport of lactose into the cell. And the function of acetylase, here it has been said that they remain a mystery, but the main function of this acetylase is to transfer an acetyl group from acetyl coenzyme A to galactosides, glucosides, and lactosides. So the function of this galactoside acetylase is to transfer the acetyl group from acetyl coenzyme A to the galactosides, glucosides, and lactosides. Now, what are galactosides, glucosides, and lactosides? Galactosides is a glycoside which consists of only galactose. Glycosides are actually what? They are molecule which has a sugar bound to another sugar by glycosidic bond. So galactosides here refers to only the galactose sugar is present. Okay, in case of glucosides only glucose is present attached by the glycosidic bond. So this is the function of these three. Now I want to show you the figure of this lacoperon. This is the figure of the lacoperon where you can see I refers to the regulator genes located somewhere in the upstream. Okay, it is not just like that it is located next to the promoter. It is located somewhere in the upstream, maybe 100 base pairs away or many kilo base pairs away, and that regulatory gene will produce a regulatory protein. This regulatory protein will interfere with the operon, and operon not it will interfere with the operator actually, and operator is acting just like an on off switch, turning the expression of the gene on or off. Okay, so how does it work? So repression of the lacoperon. Now one more thing I want to say, that as these genes that we are talking about, LAC-Z, LAC-Y, and LAC-A, all are encoding three different enzymes, and they are three independent translational units. Means there are three open reading frames right next to each other, and due to this nature, the mRNA in prokaryotes are known as polycistronic mRNA. Prokaryotic organisms contain large number of polycistronic mRNA that we know. So in this way, these open reading frames are located right next to each other to form this polycistronic mRNA. Now the main function. First, I will talk about how the lac operon is turned off. Is a general procedure. It is normally occurs when lactose is not present. The regulatory gene, when lactose is present, then the lac operon will turn off. The regulatory, sorry for that again. So the regulatory gene is constitutive. It is expressed at constant rate, leading to the synthesis of the lac repressor. So this regulatory protein that is found somewhere in the upstream, it is producing this lac repressor or this regulatory protein. Lac repressor is the tetramer made up of 4 subunit, and they have a molecular weight of about 150,000 daltons. This repressor, this lac repressor that we are talking about, it is a regulatory protein, right? It is specifically binds to the operator gene. See the figure. The I here refers to the regulatory gene located in the upstream. With the help of transcription, it forms mRNA, and translation, it forms that protein, the repressor protein. The repressor is made up of 4 subunits. It is a tetramer made up of 4 polypeptide chain, and this repressor will then ultimately bind to the operator to turn off the lac operon. Okay, and thus when it binds to the operator gene, it prevents the RNA polymerase to bind to the promoter site. So RNA polymers cannot bind to the promoter now because the operator gene is occupied by the repressor, and thereby blocking the transcription of the structural genes. This is what happens in the absence of lactose. So this is occurs when lactose is not present. When lactose is not present, we don't require these genes lac Z, lac Y, lac A, because there is no lactose, and these genes are only, their function is related to lactose, related to lactose breakdown, related to lactose um, insertion into the cell. Okay, related to the metabolism of lactose. But if lactose is not present, this genes is useless. So prokaryotic cell have a system to turn off this lactose related gene, lac operon related gene, by binding of the repressor to the operator. Okay, so when lactose is insufficient, when lactose is not present, then this regulatory gene, they constantly synthesize this regulatory protein known as lac repressor, and this lac repressor binds to the operator and prevents the binding of RNA polymerase to bind to the promoter region, thereby blocking the transcription of these structural genes. So you can say that here the repressor, the lac repressor molecule, acts as a negative regulator of gene expression. But what happens when lactose is present? If lactose is present, here lactose is acting as a inducer, right? Here lactose is acting as a inducer in the medium. That lactose bind to the repressor molecules. As the repressor have a high affinity for the lactose, as you can see in these writings, the repressor molecules have a high affinity for lactose. Lactose molecules binds and induce a conformational change in the structure of the repressor. If lactose is present, lactose binds to the repressor, because repressor molecule, the lactose repressor molecule have a high affinity for lactose, so it binds with the lactose. So the lactose molecule bind to the subunits, it binds to the subunits of the tetramer it binds to the tetramer repressor, and causes a conformational change in the structure of repressor. The result is that repressor gets what? Inactivated, and therefore it cannot bind to the operator gene. Ultimately, RNA polymerase attaches to the DNA at the promoter site and the transcription will proceed, leading to the formation of these transcription of these LAC-Z, LAC-Y and LAC-A, which will ultimately produce these three enzymes and will deal with the lactose. So here we can see in the diagram that the repressor has been binded with the lactose. Although lactose is not look like this, but for simplicity it has been shown that lactose bind to the repressor and inactivate the repressor, and thereby by binding to the repressor, what happened? Repressor could not able to bind to the operator region, making the lactose operon to turn on, and the RNA polymerase would able to bind to the promoter and carry out the transcription of this LAC-Z, LAC-Y and LAC-A. Now one more interesting thing that you can see here is about this CAP-CAMP complex, the catabolite activator protein. In short, it is called CAP, and CAMP is your cyclic AMP. This is also required for RNA polymerase to bind to the promoter. This we will talk about just a bit of later. Okay. So normally lactose is acting here as an inducer. It is inactivating the repressor molecules and causes the synthesis of lactose operon. Now, thus these three enzymes, galactoside, beta galactosidase, galactoside permease, and galactoside acetylase, will be synthesized. So here lactose is acting by inactivating the repressor molecules. Hence this process is known as derepression of the LAC operon. Now there is also one more category of inducer which can permanently can cause the lactose operon to turn on permanently. That is called gratuitous inducer. So gratuitous inducers are actually structural analogs of LAC operon. Means they look alike lactose, but they are not lactose. So there are certain structural analogs of lactose which can induce the LAC operon, but they are not the substrate for these enzyme beta galactosidase. So they will not be broken down. The gratuitous inducers are structural analogs of lactose, but they are not, what can I say, they are not, they are not the substrates of beta galactosidase, right? So they will not be broken down, and thus the lactose operon will permanently turn on. So such type of substrates are called gratuitous inducers. A best example of gratuitous inducer is IPTG, isopropyl thiogalactoside. It is a gratuitous inducer. It is normally used in experiments to show that how the lactose operon works. Now we come to this point of catabolite gene activator protein. Now catabolite gene activator protein is fully related with glucose. Why? It has been observed that when the cells of E. coli or any prokaryotic cells who have this LAC operon, when these cells were grown in a medium containing both glucose and lactose, E. coli cells utilize glucose first. When the glucose level get depletion, then only it will utilize the lactose. So here glucose is indirectly interfering the lactose operon. It is interfering the lactose operon. How? Normally, if I look at the figure here it is given, glucose can inactivate this enzyme. Glucose is entering to the cell and glucose is interfering the enzyme adenylyl cyclase. Normally, adenylyl cyclase converts ATP into cyclic AMP. This cyclic AMP acts as a secondary messenger. Normally, you will see them in the signaling process when you are having the signaling chapter. This cyclic AMP acts as a secondary messenger. It carries that message, it conveys that message inside the cell. So this cyclic AMP now then binds to the it binds to the cap, that means catabolite activated protein as shown in rectangle, and the cyclic AMP is shown in circle. So this cap complex form, it binds to the promoter and allows the RNA polymerase to bind. But if glucose is present, it will inactivate this adenylyl cyclase, and if it inactivates adenylyl cyclase, cyclic AMP will not form, and if cyclic AMP not form, then this cap complex will not occur. As a result, if this cap complex not occur, it will not it cannot be able to allow that RNA polymerase to bind to the promoter and causes the lac operon to turn off. So indirectly, glucose is acting as a repressor. Okay, it is repressing, or you can say inhibiting the lac operon. Okay, let us see what has been given in the written material. The cells of E. coli utilizes glucose in preference to lactose. When both of these glucose and lactose present in the medium, after depletion of the glucose in the medium, then utilization of lactose starts. So it indicates that glucose somehow interferes with the induction of the lac operon, and this is explained as follows. Attachment of RNA polymerase. Attachment of RNA polymerase to the promoter require a protein, catabolite activator protein, or you can say catabolite gene activated protein bound with cyclic AMP. This cyclic AMP is actually an unstable molecule. It does not remain for a long time. It just only to convey the message. Okay, so that at least in that small time duration of time, RNA polymers can quickly grab onto the promoter and start the transcription process. The presence of this glucose lowers the intracellular concentration of cyclic AMP. If there is glucose, it lowers the cyclic AMP levels inside the cell. How? Why? Because how it lowers the cyclic AMP concentration inside the cell? Because by inactivating the enzyme adenylyl cyclase. Adenylyl cyclase normally responsible for the synthesis of cyclic AMP from ATP. Now, during diminished level of cyclic AMP, this cap complex is low. Formation of cap complex is low. Therefore, binding of RNA polymerase to the DNA due to the absence of cap complex and the transcription are almost negligible. RNA polymerase binding will be almost negligible because there is absence of cap complex. Thus, glucose interferes with the expression of lac operon by depleting the cyclic AMP levels. This is the main line. How glucose interfere the expression of lac operon? Simply saying by depleting the cyclic AMP levels. Addition of exogenous cyclic AMP is found to initiate this transcription of many inducible operons, including the lac operons. It is now clear that, so here you can see that if this event occurs, you can also again you can increase this cyclic AMP by exogenously by inserting exogenous, means from outside we insert the cyclic AMP levels. insert the cyclic AMP levels to initiate the transcription again of this lac operon genes. It is now clear that presence of cap-camp complex is essential for transcription of structural genes in lac operon. Obviously, without cap-camp complex, you cannot able to do transcription because RNA polymers won't bind then. Thus, cap-camp complex act as a positive regulator for the gene expression. It is therefore evident that lac operon is subjected to both positive by repressor and by negative regulation. So this was all about the lac operon.